but today I get to introduce two people, and our speaker is Kelsey, who's been co-hosting, and she's a PhD candidate here at UVic. And well, I think she'll talk a little bit about herself during her presentation. And the other person to present today is actually someone who's been taking over from Kelsey. So today is the handing over of the passing of the guard, or handing over the baton, or whatever metaphor we want to use. But Eloisa will be taking over as co-host. And Eloisa is also a PhD researcher here at UVic, also studying septic systems. Eloisa, do you want to just give a quick, or introduce yourself? And you've been to all the talks, but
I don't have much to add aside from what you've already said, but yeah, my name's Eloisa, and I study the septic fields of the septic system. And I've got big shoes to fill, but I'll do my best.
Okay, so we're all a bit skeptical of what's going to happen today. So Kelsey, do you want to take it over?
Yeah, sure. Thanks. I'm just gonna share my screen first. Hope that all works. Can you see just the slides?
Yeah, just the slides.
Great. Oh, too far. All right. Yeah, so today is a little bit different for me. I'm usually not the one speaking for too long, so bear with me. But I am really excited to welcome everyone to today's talk. It's going to be a little bit different if you've come to the previous talks than what we've been used to. I'm really excited to be able to share this space with so many accomplished researchers and professionals, and just kind of share really just the beginning of my own journey kind of into the WASH space. And so, like Keitano mentioned, I am just starting my PhD, and so because of that, today I'm really going to go over just some kind of big picture motivations behind my research, dipping into my master's, my previous master's research, and my current research. But I have also highlighted a few areas to just be a little bit more interactive, and maybe get some audience participation, and also take the pressure off me, which was planned. So today's talk is going to be about all things septic and non-sewered sanitation. But before I really dive into that, I just want to take a moment and do a territory acknowledgement. We did do one at the beginning of the series, so I think it's important to also have our last session end with one. Especially for me and my research, I think equality in sanitation can only be achieved if the systems that perpetuate inequality are addressed at all levels. As a student and a researcher, I am deeply honored and grateful that I am able to share this space with these traditional hosts and original occupants of the land. And so before I get into what exactly my research is, I think it's important just to give a little bit of background about who I am and who I am in this space of water, sanitation, and hygiene. So my background, I have an undergraduate degree in environmental engineering from the University of Waterloo. So I am an Ontario transplant. I have a master's in civil engineering from the University of Victoria. I actually realized that it was last year, exactly on this day, that I defended my master's, which is cool. And currently I am a PhD student in the civil engineering department at the University of Victoria, with Keitano as my one supervisor, but I'm also co-supervised by Dr. Linda Strande from Anaerobic in Switzerland. So both my master's and my current PhD research is kind of at the intersection of environmental engineering, public health, and sustainable sanitation. My research is specifically focused on characterization of non-sewered sanitation systems, functional microbiology, so characterizing anaerobic degradation pathways in containment. Sounds confusing. I'm still confused by it, but I will go over it a little bit more in detail shortly. So again, just to give some background, I think it's important to give some context as to how I ended up in this position and this research field that I'm in. I don't think everyone grows up being a 5 or 6 year old kid thinking like, wow, I'm going to study poop for the rest of my life. So I just want to give a little bit of background. So if I kind of rewind back to when I started my undergrad degree in 2013, I definitely would never have anticipated being kind of where I'm at now. I am kind of saying that I am incredibly grateful for the opportunities that I've been able to gain access to and the knowledge that I'm acquiring through this kind of journey. And it's been both a very like physical journey, so a lot of traveling from Ontario to BC to Switzerland for an internship that kind of propelled this PhD journey. And now in the coming year, I will also be traveling to Lebanon and Uganda for fieldwork, which I'll explain a little bit later. So yes, a physical journey and also an emotional journey. And so my research resonates with me on a very personal level due to a variety of circumstances. When I was younger, my family actually had very sporadic access to sanitation and sometimes even drinking water. And we only ever had access to non-sewered sanitation systems, first with a composting toilet, and then when I was older, a septic tank system. So very familiar with the non-sewered sanitation system in a Canadian, rural Canadian context. And it's something that resonates with me quite deeply. And so I know what it's like not to have access to reliable water and sanitation. And this is something that I really bring to my research and something that I find really motivates me. And so before I kind of get into the nitty-gritty of what my research question is and all of my objectives and goals, I did just want to take some time and open it up for the audience to maybe share either out loud or in the chat some of your experiences with non-sewered sanitation, or maybe even some common myths you've heard about sanitation, since that's kind of central to today's talk. And I just want to shout out to my research lab members for their great suggestion for these questions. So if no one answers, I might call on you guys. Don't all talk at once. Louisa, what have been some of your experiences with non-sewered sanitation?
Oh, I saw Judith unmute. Did I see that right?
Oh,
no, but go ahead.
I don't have experiences with non-sewered sanitation from, a daily standpoint, but more so probably when I'm out camping and that's the only thing available. But in terms of common sanitation myths, one that I keep hearing frequently is just how frequent you should empty your septic tank. And so I feel like that's something that needs to be cleared out amongst users and practitioners.
nice. Also a bit of a pun, cleared out. Appreciate that. Thanks for sharing. I can't see the chat, so I'm not sure if anyone is posting anything in the chat, but
There's something interesting in it, though.
Yeah, we've got something there. Colin, would you like to unmute? I can also read it.
I think
Okay,
his mic isn't, yeah.
Yeah, so Colin says, I'm now studying advanced septic systems, a new technology called multi-soil layering system to provide advanced treatment of effluent from septic before discharge into open water bodies. So you've got another septic buddy.
oh, cool. That
And
sounds really interesting, Colin.
yeah, from Pablo, he shared that one myth he knows is that centralized sanitation solutions are always the most adequate solution.
That's a really good point. And I think that's kind of something I'll touch on later, and something that I think is central to all of our research, those working in the non-sewered sanitation sphere, is kind of finding that common middle ground between what sort of is the dream of sanitation, which sometimes doesn't necessarily like match the reality in terms of expectations, but also in terms of treatment. And there's a lot of good and a lot of goals that can be met with non-sewer sanitation that sometimes I think go unnoticed. So cool. All right. Well, thanks everyone for sharing. So I will maybe continue moving forward, but if there are any other thoughts that come up as I'm talking, please feel free to put them in the chat or raise your hand, and there will be time at the end as well. So minimize my face on Zoom. So for my research, now you know kind of why personally I'm interested, but why should the rest of the world care? So over 30% of the global population does not have access to basic sanitation systems. And just for comparison, only 15% of the global population lacks access to cell phones. So right now, more people can swipe than wipe. So it's still a pretty big problem and something worth looking into more. And so that kind of leads to what my research looks like. You can tell I like graphics. So the overall objective of my PhD research is to characterize anaerobic degradation pathways in containment of non-sewered sanitation systems. So what does that mean? It means that I'm looking just at the containment system itself. I'm not looking at the downstream soil leach fields. Specifically, I'm looking just in kind of like that first step. So if we use a septic tank system as an example, it would be that actual physical chamber. That's the focus of my research. If you do have questions and you really want to dive deep on the soil systems that are often paired with these containment systems, you can talk to Eloisa, because that is more the focus of her research. And so for me, what this looks like and what my key objectives are to first understand the microbial community composition within the spatial zones in these containments across different geographic regions. Secondly, I want to analyze the governing mechanisms. So how do these substrates actually get converted into their final end products? So greenhouse gases, how are these organics being converted? And then thirdly, develop an improved model for non-sewered sanitation containments that can include this quantitative field and lab data that I am hoping to generate, which will inform not only greenhouse gas mitigation strategies, but overall operation and management recommendations in globally relevant settings. So that's kind of the 2-minute synopsis of what I'm hoping to do in the next 3 years. So I'm just going to spend a little bit of time kind of digging more into that. And so that kind of brings us to the first component of my research. So, what do we really know about these types of sanitation systems? Currently, right now, there is a gap in knowledge of the in situ biodegradation processes in non-sewered sanitation, especially when you compare it to, your centralized wastewater treatment plants, your engineered bioreactors. There's lots and lots of literature and research and textbooks on those systems, but a lot of times that research and that knowledge is applied to non-sewered sanitation systems without taking into account all of the different factors that apply to these systems. So this leads to a gap in knowledge. And there's also a gap in understanding of the relationship between the measurable influential parameters for these non-sewered sanitation systems and how that affects the microbial diversity in containment. And so in order to gain insight on these active pathways, these active anaerobic pathways, or what's assumed to be these active anaerobic pathways, the first part of my PhD research and what I've spent the last 8 months preparing for is to conduct a field-based scoping study, which needs to first investigate how these different zones in containment contribute to anaerobic microbial community diversity. So a lot of words there, but essentially, if I was to represent this in a picture, this is what it would look like. And so what this is a summary of the questions that I'm trying to ask through this study. So what are the main macro scale concentration gradients of influential parameters? So talking about dissolved oxygen, temperature, oxidation reduction potential, pH. So all of these parameters that we're familiar with in the wastewater sphere, but specifically looking at them for non-sewered sanitation. And then how does the local availability of these influential parameters, so your electron donors, your acceptors, your inhibitory substances, how do they influence the microbial community? And then ultimately, what is the microbial community diversity in containment in these different spatial zones, and then also in the different spatial zones geographically as well, since we know that these systems are used not only in one location, they're used worldwide. And so what does that actually look like in terms of what I'm going to be doing? So I will be sampling in 3 different geographic locations, so Canada, Lebanon, and Uganda, with our partner organizations. And this is just I know the text is small, but this is just a photo of my grab sampler that I would be using to help me take grab samples, because essentially for the next 1.5 years, this will be my life. A lot of sampling, a lot of different septic tanks. These are some pictures from the summer of me field testing some of my equipment and sensors. And that is how I'm going to try and answer these questions in regard to these parameters and how they influence these microbial communities. So that is a really quick overview of my first sort of goal of my PhD. And this all leads really nicely into my second objective, which builds on this first objective questions, but really digs into the gap in knowledge about the actual governing mechanisms that are important for substrate conversion to final end products. And so when we're talking about substrate conversion in non-sewered sanitation systems, we're mostly talking about these anaerobic degradation pathways, since it's assumed that these wet non-sewered sanitation containments are mostly operating in anoxic or anaerobic conditions. So these are big assumptions, but to be able to gain more insight into this, there is a need for the quantification of these methanogenesis pathways to investigate these assumptions further. Currently, there's a lack in knowledge linking these in situ microbial communities to actual ecological function. So what does that mean? Basically, what it means is that we still don't have a good understanding of the causal linkages between changing microbial diversity in containment and specific dominant activity and substrate conversion. So again, what would that look like in a picture? For those of us who don't just have the anaerobic degradation pathway burned into their brain, or those who did not just take a biology final this week, this is a bit of a refresher. So when we talk about anaerobic pathways and substrate conversion, essentially this is what we're looking at. So this is a really simplified view of the acetoclastic methanogenic pathway. There are different pathways, but I'm just kind of using this as an example. So to be able to kind of dig into how these substrates are converted and what the end products look like, how much of these end products are being produced, we really need to start asking questions kind of at the crux of these arrows kind of flowing through this diagram. So for the acetoclastic pathway, one question we could ask is, how is acetate being converted into the final end products? Who is responsible for that? And by who, I mean which microorganisms? Are there other pathways that are dominant for substrate conversion? What are the factors that influence this? What are the differences spatially within the containment? Is there more activity on the bottom layer? Is there more activity on the top layer? If you go from top to bottom in your septic tank, why are these differences occurring? And so there's a lot of questions to be generated here, and also hopefully a lot of answers to come out of it. And again, I'm still at the beginning of my research, which is why I don't have any answers for you. But this is what I'm looking to tackle. And kind of moving out of that second objective and digging into those really teeny tiny microbiological worlds. This leads me to my third objective and kind of the wrapping it all together. And since, of course, this PhD is in engineering, there has to be a model. So the third objective is to look at quantifying, using this quantitative data from both the field work and the lab work to improve non-sewered sanitation containment models, specifically when it comes to this final end product conversion, which is methane, which is CO2. These are obviously quite important for greenhouse gas emissions. And so using this data to improve existing models and be able to leverage them for community scale non-sewered sanitation models as it pertains to management recommendations as well as mitigation strategies for climate impacts. And so in order to achieve this, as part of my sampling, I'm also going to be looking at in situ gas flux measurements. So essentially what that looks like is me using this really fancy piece of PVC pipe, essentially operating as a gas chamber. So I'll be able to take in situ gas measurements along with grab samples and microbial sludge samples to be able to start to characterize what those gas emission conversion rates look like and how all of these parameters work together to influence that in different rates of conversion. So that is very big picture what I'm aiming to do for my PhD. So kind of taking a step back, what does that mean for me, for my next steps? So the last 8 months, I spent a lot of time doing literature review, strengthening partner relationships, field testing my equipment, and my first field campaign will be starting in March of this year. I'll be going to Lebanon to work with our partners there, a really great team from Cubex, young Lebanese startup working in the informal Syrian settlements as well as in some urban contexts in Beirut. And I've had experience working with them in my master's through an internship. So now I'm really excited to be able to actually go and visit them and work with Rita and Charbel, who are the two folks that I have arrows on. And they're going to be working with me to collect samples and also characterize sludge from containment systems there. And before I get to some more questions and answering some questions, I just want to give some acknowledgements to both of my co-supervisors. So Dr. Linda Stronde, as you can see in the top photo, enjoying a beer after a field testing day up in the Swiss Alps. And then also Keitano, who is here today. And a lot of these graphics came from a poster that we designed earlier this year. And so, yeah, none of this would be possible without them or the support, the financial support from those various institutions. And then again, just a shout out to other researchers that I've encountered and have the pleasure to work side by side with. Super excited to pass this Wash Canada Talks over to Eloisa. Unfortunately, this was the only picture I had of the three of us. Eloisa and Keitano look great, me not so much. But I am really excited to share this space and also continue this journey with other researchers in this field. But yeah, that is all I had for my presentation. I did have this kind of question at the end just to get some feedback from other researchers about their motivations, but I do see there are like a bunch of chat questions, so I'm wondering if maybe we should pivot and maybe answer some questions. I'm open.